Chapter 15, Segment 3. Genetic Variation in Bitter. The first studies about the genetic variation in bitter were done by Arthur Fox in 1931, and his studies actually started at an accident when he was working in the lab and he spilled the compound PTC, and one of his colleagues who was working nearby noticed a bitter taste, but Fox tasted nothing. So this led to the development of non-tasters, who taste little bitterness in PTC, and tasters who perceive PTC as being bitter. And this was found to be an inherited trait, as the non-tasters carry 2 recessive alleles, and tasters have either one or both dominant alleles. So the thresholds. To study the thresholds, subjects were given 4 cups of water and 4 cups of varying concentrations of PTC, and they were asked to sort all of 8 cups, and the correct sorting determined the thresholds. And the distribution was bimodal, with non-tasters showing very high thresholds and tasters showing very low thresholds. So the next person to do research on this was Ronald Fisher, and he focused on the nutritional implication of genetic variation and suggested that tasters were more finicky eaters, and this makes sense because the bitter tastes were more intense, so they were less likely to eat more bitter foods. And then there was Dennis Drina in 2003, and he discovered the location of the gene that expresses the PTC and the PROP receptors. So next came the discovery of the supertaster, and this was a new look at genetic variation in taste. Instead of thresholds, investigators could look at suprathreshold taste and plot the psychophysical functions, showing how perceived taste intensity varies with concentration. And this led to the discovery of supertasters, and this was done through cross-modality matching. So what happened was researchers asked subjects to match the bitterness of PROP to other sensations, and obviously non-tasters matched the taste to weak stimuli, like the flicker of a match in a dark room. And tasters matched the PROPs to stimuli with slightly stronger, like a mild headache or a loud buzzing. And the supertasters likened PROP to very intense stimuli, like the most pain they've ever experienced. And then Valerie Duffy did more research on this, and she divided individuals into 3 groups: the non-tasters, tasters, and supertasters. So the combination of ability to taste PROP and the possession of a large number of fungiform papillae produces a PROP supertaster. And those with the most numerous fungiform papillae experience the most intense sensations of oral burn and oral touch as well. And this is because the fungiform papillae are innervated by nerve fibers that convey burn and touch sensations along with taste sensations. And actually what was found was that removing input from one of the taste nerves can intensify taste sensations. And this is because inputs from the different taste nerve inhibit one another in a brain, so loss of input from one nerve releases others from the inhibition. So they found that taste experience is the outcome of genetic variation, pathology, and interactions among all of the cranial nerves innervating the mouth. So next is health consequences of taste sensation, and they found that the variation in the sensory properties of foods and beverages affects food preferences and thus diet, and diet affects health. So for example, some vegetables produce unpleasant sensations to medium and supertasters, leading to less consumption, and that reduced vegetable intake is in turn a risk factor in colon cancer. So like I said, variation in the sensory properties of foods, beverages is linked to health because that variation affects what we choose to eat, and diet affects health. So next is the wisdom of the body. How do we solve the omnivore's dilemma? So the omnivore's dilemma is described as the array of choices of what to consume, or more modernly, the fact that humans need to find healthy diet among the dizzying choices available to us today. So taste receptors detect specific features of molecules, and because of this, the taste system responds to a fixed and much smaller set of molecules than the olfactory system. This is consistent with the role of the taste system for detecting nutrients and anti-nutrients before we ingest them. And each of the 4 basic tastes is responsible for a different anti-nutrient or nutrient or anti-nutrient. And Jacob Steiner did research on this and found that infants respond with the stereotyped facial expressions when sweet, salty, sour, and bitter solutions were applied to their tongues. This was found to be because these reactions were mediated by a very primitive part of the brain. So next is, how do we regulate nutrients? Early belief in specific hunger gave way to identification of conditioned preferences and aversions. So Kurt Richer was the first person to do research on this. He proposed a simple mechanism for the regulation of nutrients, and this is called the specific hungers theory. And this proposes that the need for a nutrient causes the body to crave it. And then Clara Davis did further research on this by allowing a set of infants to eat whatever they like, and she found that the infants thrived because she believed that their bodies were telling them what to eat based on the nutrients that they needed. And that, but they found that this theory seems to be limited to sweet and salty. For craving to cause an animal to seek out and take in a needed nutrients, a sensory cue would have to unambiguously be associated with the nutrient. So this works for salt and sugar, but not most nutrients. Later it was found that Clara Davis's infants did not choose a healthy diet on purpose, but rather they ate a variety of foods because they got bored eating a single food. Rosen ended the belief that the brain was hardwired to tell us what to eat, but and later found that it was actually we learned what to eat and what not to eat, and basically that we come to like or dislike foods based on the consequences of eating them, and this is called conditioned aversion and conditioned preference. So also called evaluative conditioning, which is the association of negative or positive affect with an experience.